That's it. I think that was Dominique. My apologies, everyone. So the purpose of the roundtable event was to bring together some of the experts, just to kind of talk about the landscape of aniridia research, and to maybe think about our priorities and how we can work together and collaborate for the future, for the benefit of our patients. So I think what we'd like to start with is maybe just go around the screen and just quickly introduce everyone. So the person next to me on my screen is Veronica. Over to you, Veronica.
Yes, well, so I have worked for quite a long time, as you might have gathered, on PAX6 and aniridia. I have to say I am now retired, but it is such an exciting time to be thinking about the possibilities of what we are going to be able to do, because I think there's just better management. I mean, even going back to what Tony was saying, which I have to say I never thought about before, because I probably didn't know that having much more specialized ophthalmologists working in different areas. So there's people who are experts in cornea, in retina, etc., makes a big difference. And I think patients are now getting a better deal, and I'm sure we can improve a bit more in that direction. But there are so many new technologies as well. And Maria and Julie and Neil, Cheryl, all of you actually, other than me, are working in that direction. So I'm hoping that you're going to do quite a lot of the talking.
Julie, would you like to just quickly introduce yourself?
Yes, hi, I'm Julie Daniels. I'm a scientist at the UCL Institute of Ophthalmology and work very closely with my clinical colleagues, Saj and Maria at Moorfields, and others as well here at this meeting. So yes, like Veronica said, very excited about the future. There are lots of new technologies hopefully going to be coming along to help us. And, you know, even if they don't give us the answer straight away, I hope very much from the trials that we do, we will learn from them and be able to improve therapies for patients in the future. I think there is a lot of promise, and I think now there's much more emphasis on engaging patients and what they want and how they want to help us, and I think that's going to be so much more positive. We can't, especially as scientists like me, it's very easy to just be in the lab and, you know, forget that there are people on the end of it. So I very much value my clinical colleagues as my bridge to all of you.
Neil?
Sure, yeah. So I'm doing corneal research here in Linköping in Sweden, and we are, maybe I will talk a little bit later about our cost action, but we're working with a number of different groups around Europe, trying to learn more about aniridia from various patient cohorts, but also trying to look more at a basic science level to see what's happening in the cornea. And we're really happy to collaborate, more and to share our knowledge and share our results, both positive and negative as well. I think we can all learn from this and move forward.
And then to the wonderful Cheryl Gregory Evans, who I should tell everyone was my PhD supervisor between 2005 and 2008, and one of my mentors and inspiration. So.
Yeah, hi from Vancouver. I wasn't able to join your meeting very early this morning because it was so early for me at 2-3 o'clock in the morning, so I missed some really good talks, unfortunately. I'd be interested to know if those would be available online for people to look at a later time. I'm not sure whether that's possible.
They will.
Anyway, I've been working in...
They're all recorded, so people can watch the playback.
So I've been working in inherited eye disease my entire career, but since I came to Vancouver, I was specifically involved in working with PAX6. As many of you know, we're interested in finding new therapies and atalurin was one of them. And is somewhat disappointing that it didn't reach statistical significance. But there's many reasons for that. I'm sure we'll discuss what outcome measures are and how to deliver the drug in an appropriate manner. And we're now looking at other therapies that might be relevant to patients. So continuing with our striving to find new therapies.
Sarge?
Hi, so I'm Saj Ahmad. I'm one of the corneal surgeons at Moorfields. Work closely with Maria and Julie on aniridia. I've worked in my research interest is actually limbal stem cells and development of cellular therapies for limbal stem cell deficiency since about 2003 when I did my PhD. And although historically I've worked a lot on chemical burns related limbal stem cell deficiency, ectodermal dysplasia and aniridia are the two genetic diseases that are associated with limbal stem cell deficiency. And so I look after about 120 patients with aniridia-related keratopathy. So I cannot but want to develop better therapies for them. And so hence my interest in aniridia.
Brilliant. And then to Dominique, if you would like to introduce yourself, and then I'll let you continue to just talk to us a bit about ERN initiative.
Okay. Thank you very much, Maria, for giving me the talk. So mostly I'm a pediatric ophthalmologist, specializing in anterior segment, corneal graft, and anterior segment dysgenesia surgery, and also doing research at INSERM, and so about innovative therapy or different phenotype, genotype research. And so, of course, we work together, and I really think it's really very important. And I'm very happy to work with Neil with the cost action, and that's why I just wanted to show you the ERNI, because I think it's really an interesting point. And I will share, if you mind, if it's all right, I will share my slides. If I can do that, I will try to do that. Share screen. You tell me if you see the slides.
Not yet, Dominique.
Not yet. Okay. So not yet. I will try again. Not yet. You don't see the.
No, maybe in the interest of time, because we only have 35.
Okay, so we have very few.
Talk us through it, Dominique.
I can do that for sure. And so, so ERNI is an association for the best coverage of the rare eye disease. It's an association and a coordination with Hélène Dollfuss in France. I'm Oftara coordinating. It's around 20 centers, and there is a constitution of work group of 10 work groups, like retina, neuro-ophthalmology, pediatric ophthalmology, anterior segment, low vision, genetic testing registries, research, education, and communication. And so we work in meetings, and we produce some papers together. And really the aim is to best coverage of those rare disease. And we know that there is barriers for the early diagnosis, patient management, and research. So I think it's really to join forces in the European reference network. And I think it works, and everybody of Europe, of course, we are happy that you join. So that's it.
Thank you, Dominique. And then I'm going to pass to Neil just to say a few words about the cost EU, please.
Sure. Thank you, Maria. Yeah, I would just like to, for those who are not aware of it, just to inform that we have an EU cost action that's running, and it will be running for at least 2 more years. There are to date about 30 countries that have joined, and also the United States has joined as an international partner country. So they're also now formally eligible to take part in our activities. And we arranged this action along 6 different working groups, and these groups have distinct goals. For example, there's a clinical guidelines working group which Dominique is leading, and here our goal is to come up with a pan-European consensus statement that's evidence-based for aniridia care. And then we have, for example, another group that's patient-driven research. So here we're trying to create a number of different surveys that will be sent out to patients all across Europe, because many patients and groups felt that their concerns are not being really documented well enough. So we will try to do this. Also, there's the working groups on stem cells and regenerative medicine, models of aniridia for collaborative research, and so forth. So I will put the website of our cost action into the chat. So anyone who's interested can visit the website, and then please contact the working group leader for the area that you're interested in, and you can be updated on our activities, and you can it's free for you to take part. And what's also interesting about this action is that the EU provides financial support for investigators, students to have exchanges between countries for research. So anywhere from 1 week to 3-4 weeks, you can go to a different lab, learn a technique, or you can share knowledge and so forth. And this is funded up to €3,500 per exchange. So we're really glad if people will join and propose these exchanges. So I'll put the website into the chat.
Don't you also produce quite a number of small meetings online? Because I think I've listened to them through Aniridia Europe.
Okay, yes, thanks, Veronica. That's a very important point. We do have regular meetings of the cost action and the various working groups. And if you contact either me or any one of the working group leaders, we'll put you on the mailing list so that you'll be able to take part in those, especially now when it's virtual. We have a possibility to have, invite as many people as possible to these meetings. So yes, definitely, the more, the better.
So we've put together some questions just to kind of discuss. I think I'm going to just shift around, Veronica. The first question, so a lot of you would have been here for the whole day and would have heard a lot of the talks, but one of the questions is about resources. Do you think we have enough established PAX6 models within our kind of aniridia research community? So feel free to feed in.
So I don't know who is listening and who is not, but we did have Beth Simpson from Vancouver talking about some of the mice that she's made, and I know that she has made some really clever models because she's a fantastic mouse geneticist and now has worked in aniridia, amongst other things, for quite some time. And she makes them freely available.
We also definitely have a lot of the groups from today talking about the iPSC-induced either optic vesicles or limb or epithelial stem cells. My group also has the PAX6 zebrafish. I think maybe Cheryl may have that as well. So on the basis that individual groups have these kind of models, do you think that we should now be putting together some sort of Aniridia bioresource, where we can have, to a degree, open access. So if a group is requiring iPSC cells, they can go to that and request that. Should we be considering putting patient samples into a biobank of some sort? What are the thoughts of the panel?
I totally agree and support that. We're starting. It's one of the goals of the cost action. We're starting to do that, trying to share, for example, cells and protocols. I think it's great with the Aniridia animal models, for example, what Elizabeth's doing. I think it's not only, I think the sharing of information is really important too, because it's not only the actual cell or the actual model, but it's the knowledge of what to do, when to do it, how to do it. That's really important. So I think that also has to be shared.
I totally agree with Neil and Veronica also. And I really think the association like Aniridia Europe, or for me also in France, Generis, are absolutely essential. And because we work together, and this is very, very important to reach the patients also, and the patients also can be aware of preventive actions and where to go, even if they are in an area where there is maybe nobody that really have the knowledge of the Aniridia, congenital Aniridia. So I really think, Veronica, you had a very great idea to underline this point, because we couldn't do that with that association, and this is really very important, to work together.
There's just something from Beth Simpson here saying we're working on some new mouse models that are humanized, and will be the next step. That will be the next step, and all of their mice are placed at the Jackson Laboratory for worldwide distribution.
Can I just ask, what about the ethical issues about sharing patient cells? Because certainly I know in Canada, if you take patient cells and develop cell lines, you can't go back afterwards and use them for something that you didn't already say you were going to use them for, because it's unethical. And so they don't allow us to do things with cells once a patient has given them for another purpose. So I don't know how in Europe that
No, we have a very, very strict regulation. Maria, maybe you can go on, right, if you want.
I don't. Yeah, so I speak from my experience with other kind of inherited retinal diseases. So the choroideremia community wanted to put together a similar kind of biobank of their skin fibroblasts and iPS cells. And they used Coriell Institute, which is a big repository of cells. And the researcher did contact the patients, and sometimes new biopsies were taken, and they were sent there, and that is now open access to any researcher. So I think there are ways of doing it, and Coriell has its own ethics and own consent forms. So I mean, that's something that we could look into, but I've certainly sent patient cells from the UK to Coriell for banking there, in support of the community's kind of work to have those resources.
There was also even earlier HIPSCI, Human Induced Pluripotential Stem Cell Project, partly funded by the MRC and the Wellcome, and they have a deposit, and it certainly includes, for example, a lot of and there are ciliary diseases. I've forgotten. And a lot of cell lines were made, and they are freely available because we discussed it in great detail. And well, I used to be on the Human Genetics Commission, and at the time there was a rule, that you couldn't use cells from people who had died. But a breast cancer expert also on the commission said it was ridiculous. He actually resigned because he said there's nothing that the patients want more than for us to use their cells as much as possible, to learn as much as possible. And I think that's probably true. And it would be interesting to hear if there are any patients listening who have donated cells to see what they think. I wish they could speak to us, but they'll have to put stuff into the messaging system, the chats.
Sarge, I think you wanted to say something.
I think we have so we have comparable where there's a will, there's a way. So if patients want their cells to be used to understand to understand their disease and develop therapies for their disease, especially if they're orphan diseases, then we can do it with the ethics that we have. I mean, there's another rare disease called Stevens-Johnson syndrome, which affects the cornea, which I know well, and we work very closely with our partners in Japan, Korea, India, Brazil, and we have no problems in sending. Remember, cells are different to tissues as far as regulatory processes, certainly here in the UK, go. So once a cell's taken out of a tissue, it becomes something that isn't regulated specifically by HTA. As long as they know that the tissue is being used specifically for ethically approved projects, they will understand that process. As long as there's a regulatory process that governs it, they're happy. And we have the same ethics for that project in Japan, and those same leaflets and everything else translated into English are used in the UK. So it is possible, and it is a rare disease, and I guarantee, having done this for 15 years in rare corneal diseases, that you can only make it work for patients, because ultimately that's what we're here for. Ultimately make it work if we network and pool our expertise and our experiences together. We won't find a solution on our own.
So Kelly Trout really nicely talked about the WAGA registry earlier today. Now, Veronica, you in the past, I think you set up the mutation database.
Yes.
But what about an aniridia patient registry? Or do they exist in? It doesn't exist in the UK. Does it exist in Europe or in the US, Canada?
Barbara Gassner-Kellner, I can't remember what her surname is, but she organized the big Aniridia Europe meeting in Duisburg, and she I think started such a database. I don't know whether she's listening. She's an ophthalmologist and I think quite heavily engaged in Aniridia Europe. I think the patients would like that. And I did talk about it there in Duisburg, and she said that they had already, there she is, Barbara Kassman. And I don't know how far Barbara has got with setting up the database, but I think patients really want it, and we have to have phenotype as well as genotype. And I think it would be really important to start setting this up.
I just would like to add, because in France we have a database registry, which is BAMARA, and probably the aim is to obtain a European database. And so in my center I have around 12,000 rare eye disease, of course not aniridia, but more than 340 aniridia. And this database is related to the genotype and related also with the phenotype. And so I think it could be very useful if we had that in Europe, but I really understand the barriers, and it's probably very difficult. But when we are in Europe, the regulations are quite easier, and that's why I think ERN is probably, it's a strong potential to get information and to get more important information to treat patients.
So Barbara says we make the data. They're making the database for the German patients, and Beth Simpson says that Doctors Netland and Lauderdale have something like this in the US.
And
We have it in Canada as well. So in Canada, we have a Canada-wide registry of all inherited retinal dystrophies and other conditions that would include aniridia. And it's funded by the Foundation Fighting Blindness. And you can get access to the registry by an application, and it allows people to find patients that have certain genotypes and phenotypes for whatever their clinical trial is. And it's a pretty large database. I'm not sure how many aniridia patients are in it, but it's certainly, there are some. But there are also many other inherited retinal dystrophies. So they do make it available. If you want to do a clinical trial, you can apply to have access to the patients, and then the patients can be consented if they want to join the trial or not.
So just to let everyone, at least from the UK, know, there is a national registry for rare diseases that is funded by the Department of Health. We don't automatically register patients on there. Maybe we should. Sarge, that's something we need to think about, because at Moorfields we have over 350 aniridia patients. But just to say that if we don't have these registers, when we come to the point of clinical trials and you have commercial companies looking to find patients to treat, that's the first question they're going to be asking all of us. How many patients have we got, and where are those patients? And the National Register for Rare Diseases in the UK, patients can actually self-register into. But that's certainly something that, you know, from our part, we should look into promoting more, because I think the UK is behind on that, and we tend to silo our disease cohorts.
I for pharmaceutical companies, because they are looking really for the numbers of retinal dystrophies, and so we know that. Of course.
And then maybe moving on from that, talking about clinical trials, should we be forming an international clinical consortium to collect natural history data, genotype, phenotype, longitudinal data, even deep phenotyping, just so that when these clinical trials come, we know what our outcome metrics are, depending on what the therapy is, at what age we're looking to apply them. So again, I open that up to everyone.
Okay.
Yes,
so we can work together.
Add to that if you're going to do that, it would be really good to have samples of DNA, because in the future, at least, you will want to look at the interacting genotypes and to, for example, for longitudinal studies, to see what predisposes to what. And we'd learn a lot more biology that way, because it's not going to be only PAX6. There's going to be many other genes as well, ultimately, for progression.
I agree, and I'd just like to add also that you can take it one step further, and multiple centers could be centers that would participate in like a multi-center clinical trial, just because we would want to get the numbers of patients up to a meaningful level for future potential trials.
And again, centered around that, and it may be part of COST or ERN, but should there be some sort of working group or task force to look at the outcome metrics for clinical trials?
There is another entity that could be useful, is the EVICR clinical research, clinical trials, and they are working on that. And one of the it's probably about 100 and more, like 120 centers in Europe. And so this is very interesting for natural history, because we can ask to work together on this. So what is very interesting is we have a lot of networks, and we can work together by different channels. And the interesting is to find out what is the best channel to work together.
Mm.
But it's difficult.
And I think we should also look at the inherited retinal disease space, because they are kind of 10, 20 years ahead of us. And, you know, the Foundation Fighting Blindness in the US have got inherited retinal consortiums. They have standard protocols. They have multi-centers where, you know, people will submit their data so it can be analyzed in the correct way. And maybe, you know, we really should think about that now before we get to a treatment and then are in this position and having to start from baseline.
So you should think to register all. We register all retinal disease in our center, but we register all rare eye disease. That it's really a huge work. It's funded by the health ministry, and so we have some people to do that. But it's a very big work. But I'm sure it will be very useful. I don't know when, but it will be very useful for some patients.
Maria, I was just going to say,
yeah.
Sorry, Maria, I was just going to say, on the basis of things that we've just said, having developed, you know, therapies for rare disease before, cell therapies, I've sort of learned that, as Julie has and as Cheryl probably has as well, is that you actually need to think 10 steps ahead, because you don't want to develop something knowing that it's not going to be a viable option and it's going to fall down at the last hurdle, because the ultimate aim is to get a therapy that can be given to patients everywhere, or as often as we can. And we have to think, have we got the right preclinical models, which is what you were mentioning before? And actually you need both, you need animal models, you need stem cell based models, and you need adult ex vivo models. And then the next step is we need to somehow, pharma is not as engaged in certainly corneal disease as it is in various other retinal diseases, for example. But there is an incentive now, because orphan diseases are given priority as far as regulation goes, and we've heard that through, you know, the issues with COVID vaccination recently, how things can go through a different pathway if there is a clinical need. So, and orphan diseases would go down that route, and aniridia would. So, and then the next step is really to try and determine what the production of the drug's going to be and how it's going to be administered, and they're all things that we can only do if we're seen to work together.
Yeah, so I you know, that was a very important point. And Neil, in your talk, you very nicely listed potential future therapies. So one of the questions I want to ask everyone, and I'm really pleased to welcome Daniel back, what would be the top research priorities for aniridia?
If I can jump in, I would just say, I guess we should probably have a multi-pronged approach, saying there are things we could do right now for patients, or there are also things that are more preclinical, at a preclinical stage, and I think both are needed. In terms of therapies, you know, there are a lot of approved therapies that can be tried in aniridia. But there are also new up-and-coming therapies that need the preclinical validation.
Yes, and I think that as soon as we will show that repurposing compounds are not toxic, we must go on. I know it's not easy, but we should jump, and I will try to convince some people to go forward. We are very motivated, for sure, because we know patients.
Cheryl, can I? Oh, sorry, I was gonna go ahead, Dominique, and then maybe Cheryl after.
Okay, I will go very quickly. I think we are just seeing emerging genic therapy in retinal disease, and I'm sure we will have the same in the corneal disease. So aniridia is one of these. But what is really exciting is we have a lot of possibilities when we see how many talks and how many different ways we can do to improve it. So I think we have hope to do something good. So we have to wait because it takes time. But I think it could be one of or more therapies that could be effective, and maybe some could be combined. So we have to go away to go there.
Sure, I agree with all of you that particularly if you're looking at topical treatments to treat the corneal disease, it's really accessible. But the issue with it is getting the drug companies to do the toxicity studies. For most of those sort of repurposed drugs, they've been tested sort of, the pharmacotherapeutics have been tested sort of systemically. But the FDA, and I don't know about the EMA in the UK, but the FDA only allows 2 companies to do the ocular toxicity. And it's a rabbit protocol, and it costs $1 million. And unless the companies are willing to fork out the money to do that, they won't do delivery topically. So you've got to, first of all, get over that barrier, certainly in North America. I don't know about in Europe, but using repurposed drugs that haven't actually originally been tested in the eye is a big barrier. And that's why PTC Therapeutics didn't do the delivery, which would have been the better approach for atolone via the eye drop. And they were hoping to get some positive results, and if they had reached statistical significance, then they would have put the $1 million in to get the testing done in the eye. So I think that's one of the barriers that we have to sort of overcome with these. But I'd be interested to know how that's held in or dealt with in Europe.
Okay, so I think we are pretty much in agreement that it would be really useful to have these patient registries with full details and, if possible, even actual samples for DNA purposes in the long run. But I do agree with Sarge that one has to think long term, because setting up these databases registries databases is a lot of work, and you don't want to waste that work by not thinking through what you're going to need in 10-20 years time. Might not be very easy to think about that, but some databases have managed. We have the Children of the 90s database, and they had to go, which is mother parents and children born in 1990 odd, and they were all collected in Bristol, and fantastic work has been done to them, but they did have to go back and bleed them, and they made lymphoblastoid cells retrospectively, and they may well have been making iPS cells. I don't fully know, because I don't follow all their papers, which are myriad, and it is a resource that is fantastic for other people, and as you know, any resource can, for example, be used as a control in some other cases. That was one of the Wellcome Trust approaches when we first started doing genome-wide association studies, that they used one disease as a control for another, totally unrelated one. So I think we need to think about this, because I think we need to get funding, and we need to organize ourselves properly to start executing this work. So I think there is maybe just about time for a short discussion on outcome metrics for trials and actually experimentation. How do we know whether there is improvement, measurable improvement for treatment for corneal disease or limbal cell problems? I mean, is there any agreement? How do we set these standards? Who sets the standards?
Veronica, I think there's clear criteria now for understanding outcomes of limbal stem cell therapy. We know that there are systems looking at corneal haze, vascularization, quadrants of vascularization. So there are they might not be specific to aniridia, but there are ones that we can utilize from other types of cell therapy. So, for example, we were asked exactly the same thing when we developed cell therapies for chemical burns. What are the outcomes that you're going to look at? Because just as Cheryl's said in the discussion, is that the FDA often looks for visual improvement. And we know that actually the first step of a limbal stem cell therapy is actually not necessarily improvement of vision. It's actually preparing the eye for a corneal transplant that will then be successful. So the outcome is reversal of limbal stem cell deficiency. And that should be your primary outcome measure. Your secondary outcome measure should always then be vision. But that may be as a result of the cell therapy or whatever therapy, with or without a corneal transplant.
I think I agree with that. And I think one potential marker that could be interesting to look into is looking at the inflammatory cytokines in the tears, because this could be, telling you whether you're still in that chronic wounded state or not after therapy.
Yeah. Yes. I mean, there are all sorts of proteins that one can now measure with ELISAs and so on with considerable accuracy, and there are relatively cheap techniques as well. So I think that would be really good. It isn't only DNA; it can be proteins as well. And of course, Neil, you described in detail how you classified your cases. And do we have other? We've been talking about the eye because you're all ophthalmologists and eye researchers, but we do have to worry about what sort of things need to be done for obesity, diabetes, etc., sleep. And I guess we're not going to be able to discuss that here, but we mustn't forget it when we're thinking back to this discussion. I think we're pretty near the time when we should be handing back to Dave Mackay. Are there any last thoughts?
I just want to say thank you to everyone. Thank you for all the amazing speakers as well. Thank you for all of you joining the panel and to the audience for all your comments and input.
Thank you.